Dear class, this week we're going to talk about pituitary and thyroid. The neuroendocrine system, which is controlled by pituitary and hypothalamus, coordinates body function by transmitting messages between individual cell and tissue. This contrasts with the nervous system, which communicates locally by electrical impulses and neurotransmitters directed through neuron to other neuron or to specific target organs such as muscle or glands. Nerve impulses generally act within milliseconds. The endocrine system releases hormone into the bloodstream, which carries these chemical messengers to target cells throughout the body. Hormones have a much broader range of response time than do nerve impulses, requiring from seconds to days or longer to cause a response that may last for weeks or months. The hormones secreted by the hypothalamus and pituitary are all peptides that act by binding to specific receptor site on their target tissue. The hormones of anterior pituitary are regulated by neuropeptides that are called either releasing or inhibiting factor or hormones. These are produced in cell bodies in the hypothalamus, and they reach the cell of pituitary by the hypophyseal portal system. The interaction of releasing hormones with their receptor result in the activation of genes that promote the synthesis of precursor protein. The protein precursor then undergo post-translational modification to produce hormones which are released into the circulation. Each hypothalamic regulatory hormone controls the release of specific hormone from the anterior pituitary. The hypothalamic releasing hormone are primarily used for diagnostic purpose to determine pituitary insufficiency. Although a number of pituitary hormone preparations are currently used therapeutically for specific hormonal deficiency, most these agents have limited therapeutic application. Hormones of anterior and posterior pituitary are administered either intramuscularly, subcutaneously, or intranasally, but not orally, because their peptidal nature makes them susceptible to destruction by the proteolytic enzyme of the digestive tract. Corticotropin releasing hormone is responsible for synthesis and release of peptide proopiomelanocortin by the pituitary. Adrenocorticotropic hormone ACTH, or also known as corticotropin, is a product of post-translational processing of this precursor polypeptide. Normally, ACTH is released from a pituitary in pulses with an overriding diurnal rhythm, with the highest concentration occurring at approximately 6 a.m. and the lowest in the late evening. Stress stimulates its secretion, whereas cortisol, acting via negative feedback, suppresses its release. The target organ of ACTH is the adrenal cortex, where it binds to specific receptor on the cell surfaces. The occupied receptor activates G protein-coupled processes to increase cyclic AMP, which in turn stimulate the rate-limiting step in the adrenocorticosteroid synthetic pathway from cholesterol to pregnenolone. This pathway ends with synthesis and release of the adrenocorticosteroids and adrenal androgens. The availability of synthetic adrenocorticosteroid with specific property has limited the use of corticotropin, mainly to serving as a diagnostic tool for differentiating between primary adrenal insufficiency, Addison's disease associated with adrenal atrophy, and secondary adrenal insufficiency caused by inadequate secretion of ACTH by the pituitary. Toxicity are similar to those of glucocorticoid and include osteoporosis, hypertension, peripheral edema, hypokalemia, emotional disturbances, and increased risk of infection. Somatotropin is a large polypeptide that is released by anterior pituitary in response to growth hormone releasing hormone. Secretion of growth hormone is inhibited by another pituitary hormone, somatostatin. Growth hormone is released in a pulsatile manner with the highest level occurring during sleep. With increasing age, growth hormone secretion decreases, being accompanied by decrease in lean muscle mass. Somatotropin influences a wide variety of chemical processes, for example, through stimulation of protein synthetic processes, cell proliferation, and bone growth are promoted. Somatotropin is used in the treatment of growth hormone deficiency or growth failure in children. Somatotropin is also indicated for growth failure due to the management of age wasting syndrome and growth hormone replacement in adults with confirmed growth hormone deficiency. Although the half-life of growth hormone is short, about 25 minutes, it induces the release of IGF type 1 from the liver, which is responsible for subsequent growth hormone-like actions. In the pituitary, somatostatin bind to distinct receptor SSTR2 and STR5, which suppresses growth hormone and thyroid stimulating hormone release. Originally isolated from the hypothalamus, somatostatin is a small polypeptide that is also found in neurons throughout the body as well as in the intestine and pancreas. Somatostatin therefore has a number of actions. For example, it not only inhibits the release of growth hormone but also that of insulin, glucagon, and gastrin. Octreotide is synthetic octapeptide analog of somatostatin. The injectable solution and the depot formulation suppress growth hormone and IGF type 1 for 12 hours and 6 weeks respectively. They have found use in the treatment of acromegaly caused by hormone-secreting tumors. An analog of human growth hormone that has polyethylene glycol polymers attached, pegvisomant, is being employed in the treatment of acromegaly that is refractory to other modes of surgical, radiologic, or pharmacologic intervention. It acts as an antagonist of growth hormone receptor and results in the normalization of IGF type 1 levels. Gonadotropin releasing hormone, also called gonadotropin, is a decapeptide obtained from hypothalamus. Pulsatile secretion of gonadotropin releasing hormone is essential for release of follicle stimulating hormone FSH and luteinizing hormone LH from the pituitary, whereas continuous administration inhibits gonadotropin release. A number of synthetic analogs such as loprolide, goserelin, naferelin, and histrelin act as agonists at gonadotropin releasing hormone receptor. These are effective in suppressing production of gonadotropin when administered continuously and thus are effective in the treatment of prostatic cancer, endometriosis, and precocious puberty. Prolactin is a peptide hormone similar in structure to growth hormone and is also secreted by the anterior pituitary. Its secretion is inhibited by dopamine acting at D2 receptor. Its primary function is to stimulate and maintain lactation. In addition, it decreases sexual drive and reproductive function. The hormone binds to a transmembrane receptor that activates a tyrosine kinase to promote tyrosine phosphorylation and gene activation. There is no preparation available for hypoprolactinemic condition On the other hand, hyperprolactinemia, which is associated with galactorrhea or hypogonadism, is usually treated with D2 receptor agonists such as bromocriptine and cabergoline. Both of these agents also find use in treatment of pituitary microadenoma and hyperprolactinemia.